Cells are the basic units of every living organism. They can be divided into two types, prokaryotic and eukaryotic cells. Usually, bacteria and archaea consist of prokaryotic cells, while animals, plants, fungi, and protists are made up of eukaryotic cells. Today, we'll explore the structure and function of the basic components of eukaryotic cells. At the boundary of each cell is the plasma membrane, composed of phospholipids. It functions as a selective barrier that allows the passage of nutrients that service the entire cell. How large the plasma membrane is determines how quickly and how much can cross into and out of the cell. And therefore, the ratio of the surface area of a cell compared to its volume is critical. Inside all cells is a semi-fluid, jelly-like substance called cytosol, in which all of its organelles float. Don't confuse this with the cytoplasm. The cytoplasm simply refers to the interior of the cell.
Most eukaryotic cell DNA is stored inside the nucleus, the largest and most visible organelle in a eukaryotic cell. That's the main difference between eukaryotic and prokaryotic cells. Prokaryotic cells don't have a nucleus and their DNA just floats around in the cytosol. The nucleus is bounded by a double membrane of phospholipids, which we call the nuclear envelope. The nuclear envelope is perforated with around 1,000 pores. At each of these pores, an intricate protein structure called a pore complex regulates the entry and exit of genetic material and proteins. On the inside of the nucleus, a net of protein filaments called the nuclear lamina maintain the nucleus's shape by supporting the nuclear envelope. As for the storage of genetic material, DNA is organized into discrete units called chromosomes. Each chromosome is composed of one long DNA molecule associated with proteins, some of which assist with coiling DNA. This complex of DNA and protein is referred to as chromatin.
Looking at an image of the nucleus, you'll notice a dark dot. We call this region the nucleolus, which is where ribosomes are synthesized. But what are ribosomes? Ribosomes are complexes of protein and ribosomal RNA. Their job is to synthesize proteins according to instructions from the nucleus. We can further divide ribosomes into two groups, free and bound ribosomes. Free ribosomes float around in the cytosol. Proteins synthesized by free ribosomes tend to stay and do their work in the cytosol. Meanwhile, bound ribosomes are attached to the outside of organelles, such as the nucleus or the endoplasmic reticulum. Proteins synthesized by the bound ribosomes are usually inserted into the plasma membrane or exported out of the cell. Cells that specialize in secreting proteins usually have higher numbers of bound ribosomes.
Moving on, the endoplasmic reticulum, or ER, is an extensive network of membranes that accounts for more than half of the total membrane in a cell. There are two distinct regions to the ER, the smooth and the rough. The smooth region of the ER is, you guessed it, smooth, and its surface lacks ribosomes. Its main function is to synthesize lipids, including phospholipids and steroids. The smooth ER is especially prominent in the cells of hormone glands because of its role in synthesizing steroid hormones. On the other hand, the rough ER is studded with ribosomes and appears rough under a microscope. Just like the smooth ER, it also serves as a membrane factory for the cell. However, its main function is to synthesize proteins that are secreted out of the cell. For example, insulin is synthesized in the rough ER of pancreatic cells and then released into the bloodstream. To transport secreted proteins out of the cell, a sac of membrane called a transport vesicle carries it into the Golgi apparatus.
The Golgi apparatus modifies and packages secretory proteins before their secretion out of the cell. Its structure is similar to that of a stack of pancakes, except each pancake is a flattened membranous sac. The Golgi has two distinct ends, a cis end and a trans end. Transport vesicles from the rough ER merge into the cis end and drop off their products. These products are modified while they travel through each pancake, with each pancake having unique teams of enzymes that are specialized to modify certain things. Eventually, the modified products emerge from the trans end in transport vesicles, and they are delivered to the appropriate location. This was definitely a really dense video, and part 2 is coming out soon. Feel free to email me if you have any questions. Thanks for watching, and I'll see you next time.